Welcome to Science Unplugged, the podcast where science and conversation collide. Join us for a deeper look into the world of innovation. We are your hosts, Charvi Trivedi.
And Taslima Farooq. Let's explore the insights from the world of science. From the lab to the field, we'll dive into the stories and experiences of researchers, experts, and innovators who are shaping our understanding of the world.
Today we have with us Dr. Shweta Godavarti, who has done her PhD in neuroscience from National Brain Research Center. She is currently working as a research assistant professor at University of Pittsburgh. Her impressive research profile reveals a broad range of experience in the field of neuroscience. So let us begin with the basics. What does a day in your life look like, ma'am? So what exactly is the role of a research assistant professor? And like it's something we have heard for the first time. So we would really like to know what your day at the office looks like.
Sure. So just to put my position in context, so here in academia, there are three ways that you can be a part of the academics in the university. So you have something called as a teaching faculty. So these are, again, assistant, associate, and full professors, but who are fully dedicated to only teaching. So they'll be teaching the subject area of their expertise to undergraduates and graduate students. Then the second stream is that of tenure track faculty. So these are faculty who do both teaching as well as research. And so the advantage of having such faculty who do research as well as teach is that they can incorporate the latest findings from their labs and that they get the information that they gather through interacting with others into their teaching modules. So that way the students get the latest developments in science. And the third stream is research faculty. So I'm a part of the third stream. So the research faculty just does pure research. And the idea with this is, so my day is usually involves, of course, doing experiments myself, as well as I have students, undergraduates, who I am training to conduct experiments and run projects on their own. And of course, all these experiments and projects need funding. So an important aspect of my duties is also to secure grants, funding through funding agencies, either NIH, NSF, or even private institutions and nonprofits, to seek out and secure grants to be able to support my research. So that's broadly what I do.
Now, let's backtrack a bit, and I would like to know more about how you got into this neuroscience and what did your PhD work involve, and what got you here at this point. Can you please tell us that?
Sure. So I my father is a academician, so he was a botanist. He got his PhD in botany from the agricultural from Lucknow, and so I grew up in an environment where research and science was sort of like my environment, and so I was always interested in the idea of research. It seemed like it's you feel like you're a small detective who's trying to solve a mystery, and so that's something that I found very interesting growing up. So I was like, oh, this is a cool little mystery, and let me see. My mother used to buy me books like Tell Me Why and Encyclopedia Britannica, and these would have like very interesting. So if I had any questions, like for example, why does the moon move with us along with us as we are driving in a car, right? So I just go to the Tell Me Why, and it usually would have an answer to all these very very simple questions that would crop up in my mind. So in that sense, I always had a nice scientific and rational environment around me growing up. The other major influence in terms of my interest in science was when my aunt, who was then doing her PhD in molecular biology, she took me to her institute. I was visiting her for summer holidays, and I was probably disturbing her or not allowing her to do her work in the lab. So she propped me onto a stool with a microscope and put a bacteria plate underneath. And she was like, look into this. And what I saw through that microscope just blew my mind. Like you take the plate out and it's completely empty, and you put it under the microscope and suddenly there's life teeming inside it. So that's how I got hooked to research itself. And of course, there was a lot of like my mother was a huge inspiration in just getting me to pursue this academic life that I seemed to be showing some potential for. Another influence was from our school. We were taken to a lecture that was then given by Professor Mashelkar, who was a very famous chemist from India. And it's just listening to him talk about what science is like and what scientific life is like was a huge inspiration. And then I also had the good fortune to grow up in the times of Dr. Kalam when he was our president, and he would do these regular interactions with school children. You just gather a bunch of children and give a talk on, like ranging across various topics. But one of the most important topics that he was very passionate about is how science and research can be used to shape our country. So that was another big inspiration for me. So that's how generally I rolled into research, science, and academia. In terms of neuroscience, I would say I rather stumbled into it. I knew I always wanted to do something in biology, but I wasn't really sure of what exactly. So I did my bachelor's in biochemistry. And then when I was looking for master's programs, I was looking at various different programs. And it was my mother actually who suggested that this program that was being offered by National Brain Research Center sounded very interesting because neuroscience, even at that time and still now, continues to be one of the cutting edge fields in biology. And she was correct that this should be the area that I should be in, where there's a lot of scope for research. And so that's how I joined National Brain Research Center on her advice. So I joined NBRC as an integrated PhD student. So I basically did my master's and my PhD in neuroscience. To come to your question about what my PhD work was, so I worked with Dr. Nihar Ranjan Jana. So he was my master's and my PhD advisor. And our lab was broadly studying protein aggregation, which is basically when proteins are misfolded and they're not degraded. So normally misfolded proteins are degraded. So that's how the cell clears up the system. But sometimes what can happen is these misfolded proteins, either because the protein degradation system is not working properly, or there are too many misfolded proteins and they overwhelm the protein degradation system, they accumulate in the cell. And so this in turn, like this is a huge burden to the cell and can cause cell death. And this is especially, you would have heard of these protein aggregation disorders in neurological diseases, like, for example, Huntington's disease and Parkinson's disease. So the work of the lab was to try and understand why are proteins aggregating and how can we clear these aggregated proteins. So for my master's, I basically looked at Huntington's disease, and we basically identified one particular protein called UBE3A, which is a ubiquitin ligase E3A. So what UBE3A basically does is that it is part of this ubiquitin proteasome system. As the name indicates, it's composed of a lot of different proteins that come together to break down and clear these misfolded Huntington proteins. And what we found is that usually in absence of UBE3A, or when UBE3A, the protein, is not able to function properly, then you have accumulation of Huntington in the cells, and which in turn leads to Huntington's disease in the animal. So in my PhD, I basically continued to work on UBE3A, but we identified a very different role of UBE3A. So till that time, UBE3A was thought to be a part of this ubiquitin proteasome system and involved in protein degradation and clearance. But as part of my PhD work, we showed that UBE3A has a completely different role as well in the cell, and it functions as a co-activator for glucocorticoid receptors. So just putting it very simply, it helps in glucocorticoid signaling. And there is a specific neurodevelopmental disorder called Angelman syndrome. It is a type of autism spectrum disorder, and that is associated with the patients actually lack a copy of UBE3A in their brain. And what I found was that this, using animal models, mouse models specifically, that if you lack UBE3A in the brain, then you have altered glucocorticoid signaling. And glucocorticoids in the short term, acutely, are really good. Like they are what are responsible for your flight or fight response, right? When there is a stressor, you have to respond appropriately to it. So it's important in the short term, but if you are continuously exposed to high levels of glucocorticoids over a long period of time, that causes issues like, for example, anxiety, and that is detrimental to your normal function. Right. And so what was happening in these animals that lacked UBE3A is they were having very high levels of glucocorticoids that was in turn impacting their brain function and therefore their behavior. They were showing high levels of anxiety. This has since then been now documented in patients with Angelman syndrome, that they also do show high levels of anxiety, and so therefore treatment with anxiolytics has now been shown to be beneficial for patients with Angelman syndrome. So I'm glad that the fundamental work that I did has now had some impact on treatment strategies for these patients.